We're doing our presentation on the possibilities of bringing back Neanderthals through cloning. The basic form of cloning that we will be looking at is nuclear transplantation. Nuclear transplantation contains two general types. Reproductive cloning, which is where the nucleus of an egg cell is removed and replaced with the DNA from a mature cell of another organism. This modified egg is then implanted in a surrogate mother, who then gives birth to an organism that is the exact clone of the organism whose DNA was extracted. The other type of nuclear transplantation is used in therapeutic cloning, which has the same general process, but the egg is never implanted into a mother. It is rather grown in a test tube, and then stem cells are harvested to produce organs for the patient who donated the DNA. As for what we've done with Neanderthal DNA and sequencing, we first started trying to sequence the DNA for the Neanderthals' mitochondria. However, in 2005, a coalition of scientific laboratories got together to try and sequence the entire Neanderthal genome. This project was completed in 2010. Because of this project, we were able to discover that Neanderthal DNA makes up 1 to 4% of the human genome. During this process, human and chimp genomes were used as references for the Neanderthal genome. As for the process of the sequencing process requires you take 5 separate samples and sequence each of them 30 times. The process of acquiring these samples sadly destroys bone fragments, which for Neanderthals is a very large issue as we do not have many. The same process was completed on mammoths in 2007. However, this process is much easier for mammoths as their bones are both larger and more common than those of a Neanderthal.
Yeah, and kind of going off of the idea that doing this process with mammoths is easier. You have with these with the sequencing process and taking these samples, the first genome draft is going to have errors, and getting a completely accurate DNA sequence requires the process that Liam described. So an alternative option that was offered up would have you resequencing areas of the genome that's most prone to errors, and you would do this couple dozens or even hundreds of times, and this would make that first genome draft more accurate. So eventually they were successful in sequencing the Neanderthal genome, and what you get now are the possible methods of cloning. So one method is through a surrogate mother. So how you do this is step 1 is sequence the Neanderthal genome, which has been done. Step 2 would be to chop the genome up, and what this is essentially doing is then you take these the chopped genome and synthesize it into a human stem cell. And in doing this, you develop a stem cell line that eventually gets closer and closer to the sequence of a Neanderthal. This is a process described by George Church, who's a Harvard professor of genetics. And when you do this, the idea is that you wouldn't have a single Neanderthal cloned. You would create a cohort of sorts, so as to not isolate that Neanderthal, and which would have negative mental effects. So then you also have the idea of these mini Neanderthal brains. Now this is not cloning a full Neanderthal. This is just a step towards cloning the full Neanderthal. And what it is using CRISPR-Cas9, you take human stem cells, and using chemical triggers, they're coaxed into neurons. And before this, you have these human stem cells edited so that they are closer to the ancient Neanderthal version. In either way, so these are again using chemical triggers. They're turned into neurons, and which spontaneously clump together. And in doing so, they self-organize. However, because they don't have any sensory input, they're very haphazard in their self-organization. They're going to vary wildly from one blob to the other. And these blobs are lacking thoughts or feelings. However, they have synapses and fire off electrical impulses, which can be studied as they replicate some of the impulses or basic structures of an adult Neanderthal brain.
If we were to clone a Neanderthal, we'd have to consider both the social and legal implications of this and resolve those issues. Legally, there would be issues because a Neanderthal is not truly human, but also not technically an animal. So the laws that would apply to both humans and animals would be unclear. For Neanderthals would be unclear. And until these laws are made clearer, Neanderthals could be subject to mistreatment. Socially, a Neanderthal would encounter problems such as livelihood and fitting in. Although they are intelligent, based on what we do know about them, it seems unlikely that they would be able to fit in with human society. It would be unfair to task a Neanderthal with surviving in the modern world. To solve issues of livelihood, a Neanderthal would have to live a celebrity-like existence, which is normally not a happy one. So to answer the social issue, it would be to modify Neanderthal intelligence so that they would be able to live within modern society. But then what will be the point of even cloning them in the first place? A way to resolve this so that it would be ethical to clone a Neanderthal, socially at least, is to give them a protected reserve, which could be sanctioned by a government. But this is still far off from being a reality. The reality is that we do not have the technology to clone Neanderthals yet. So in the cloning of Dolly, there was 277 trials, of which only 29 embryos, among which one birth survived. Dolly, which was a survivor, had a lot of health issues. She had to be euthanized at only 6 years old for a lung disease that was caused because her cells were rapidly aging. And this degenerative cell disease was spreading to the other sheep. The Pyrenean ibex, which was an extinct species, which was attempted to be cloned. So there was 439 cloned embryos, of which 57 were able to be transferred into surrogate mothers, and of which 7 resulted in pregnancies, of which only 1 was actually able to be born, but that newborn clone died after 7 minutes as a result of lung deformities. Due to how badly this attempt at cloning went, and due to how recent this Pyrenean ibex DNA was, it would be much more unrealistic to clone Neanderthals, and scientifically it would be unjust. So in conclusion, not to mention the ethical and social issues, it is scientifically unjust to clone a Neanderthal. The failure rate is just too high, and there are too many unknowns. And furthermore, bringing back an extinct species could cause problems for us because there could be some sort of infectious disease, such as the infectious lung disease that Dolly gave to the other sheep. On the other hand, we could give the Neanderthal a contemporary illness as well. So in conclusion, the science is just not there yet to ethically do so.